Assalamualaikum, hi, my name is Muhamad Yamin and my student ID is 2019291408. Today, I'm going to present to you about my final year project titled Cytotoxicity Studies of Powdered Energy Drinks by using plant bioassay under the supervision of Dr. Lianawati Zuran Ahmad. The background of the study. Energy drinks are mainly used by the people to strengthen their physical capability, help to lose weight, and quick energy fix. The main ingredient of energy drinks are caffeine, guarana, amino acid taurine, carbohydrates, and a variety of vitamins. The aim of this study is to evaluate the possible toxicity effects of powdered energy drinks. One of the tests that can be used is by using plant bioassay. This is because plants has a very sensitive characteristics, very economical, manageable, and can be stored easily. Usually, the chromosome is in a good condition, and the most important aspect is it has a positive correlation with other test systems. Problem statement. Consuming powdered energy drinks may come with benefits, but it is necessary to study about the adverse effects of powdered energy drinks. Jenner Tanams and Lawrence had performed questionnaire on 120 students from Malaysian Management and Science University, and the result is 83.3 students were energy drink consumer. This is why more depth research about powdered energy drinks or energy drinks generally is important. One of the possible health risk factors is the excessive caffeine amount and the other substances that are unusually can be found in the food chains. Although with all the possible health risk factors caused by powdered energy drinks, there are only a few research that related with it. This is why this study is crucial to observe the effect of powdered energy drinks on the morphology and cytotoxicological change of Vecna radiata. Objective. There are two objectives of this research. The first one is to observe the effects of powdered energy drinks on the morphology of Vecna radiata, and the second is to observe the cytological changes on Vecna radiata after 24 and 48 hours of the exposure to the powdered energy drinks. Method. First, the mung bean were germinated for 24 hours in water. Then, the mung bean were exposed to the sample solution, which is extrajoss and curcuma, at different concentration, which is 1.3% and 2.67% at different hours, which is 24 hours and 48 hours. The experiment was done 10 times to use two different brands at two different concentrations and for 2 different hours. Includes controls. Then the macroscopic parameter, which is the growth rates, were observed and measured. Next, the slides were prepared by using squashing methods. Then the microscopic parameter, which is the metatic index and the percentage of abrasion, were observed and measured. Lastly, the data that collects and calculates were analyzed by using two-way ANOVA. Then the detail for squashing methods is: first, 2 cm of the root tips were harvested from the mung beans. Then the 2 cm of the root tips were fixed in carnauba solution for 50 minutes. Next, the root tips were immersed in cool water for 10 minutes before being dried with tissue papers. Then the root tips were immersed in preheat 1N hydrochloric acid for 15 minutes. Afterwards, the root tips were immersed again into cool water for 10 minutes and dried again with tissue papers. Then the 2 cm root tips were cut to 0.5 cm and it were put on slide and stained with acetoarsine. After that, the slide carefully covered with cover slips and were squashed by using thumbs to give vertical pressure. Last, the slide were observed under the light microscopes and images were taken for further analysis. Result. There are 3 things that being observed and measured. For macroscopic parameter is the growth rates, while for microscopic parameter are the metatic index and percentage of abrasion. This graph is the result of the growth rates after 24 hours being exposed to the sample solution and controls. The result shows huge difference of growth rates between controls and exposed to the sample solutions. Not only that, it showed that the sample solution for brand extrajoss have a lower growth rate than curcuma for both concentration. From this graph, it can be concluded that the concentration have a negative impact to the growth rate for both brands. Next is the result for the growth rate after 48 hours being exposed to the sample solution. The result shows that there is a huge difference of growth rates between the controls and the sample solution. The sample solution for extra jaws has a lower growth rate at 1.33 concentration, but it has a higher growth rate at 267% concentration. This can be concluded that other than the concentration that has adverse effect to the growth rates, the duration of exposure to the sample solution can affect the growth rates positively. For the discussion of the growth rates, a study by Nell stated that high concentration of chemical can inhibit or alter the plant growth. While for this research, the result shows that growth rates of mung bean are decreased with the increase of concentration, but the growth rates still have a little increase with the increase of period of exposure to the sample solutions. This means that both of these products may contain high concentration of toxic chemical because there is obvious inhibition to the growth rates when it be compared with controls, especially when the exposure time is longer. Then the result shows significant difference of the metatic index between the controls and the sample solution after 24 hours. When comparing between the two brands, Estrajos has a higher metric index than the Cucubima for both concentrations. Based on the result, it can be concluded that the concentration has a negative effect to the metatic index for both brands. Next, for the result of the metatic index after being exposed for 48 hours, it shows that the control has almost the same metatic index with 1.23% concentration for both samples. However, there is a big fall of metatic index for 2.67 concentration when compared with 1.33% concentration. Although both have almost the same metatic index for 1.23% concentration, but Estrajos has a higher metatic index than Cucubima at 2.67% concentration. Based on the result, it can be concluded that other than the concentration that has adverse effect to the metatic index, the duration of exposure to the sample solution can affect metatic index positively. Discussion of metatic index. Demutu has stated that metatic index can be used to measure the lethality of chemicals and indicates the cytotoxicity effects to organisms. This study showed that the concentration of sample solution affect the metatic index adversely because the increase of concentration will decrease the metatic index, but the increase of period of exposure will increase the metatic index. The combination of the study indicates the presence of toxic ingredient or chemicals. It is because this study shows that the sample solution caused the drop of metatic index, and the further increase of concentration will cause further drop of metatic index. Result of percentage of abrasion. The result shows significant difference of percentage of abrasion between the controls and the sample solution after 24 hours. It showed that there is 0% of abrasion for controls, and then there is a huge increase percentage of abrasion after exposure to the sample solution for both brands. Although both brands has almost the same percentage of abrasion at 1.23% concentration, but Cucubima has a high percentage of abrasion at 2.67 concentration compared to Estrajos. It can be concluded that the concentration has a positive effect to the percentage of abrasion for both brands. Next, for the percentage of abrasion at 48 hours, the controls they have 0% of abrasion, and exposure to the sample solution gave a huge increase of percentage of abrasion. Estrajos has a slightly higher percentage of abrasion at 1.23% concentration, while Cucubima has a slightly higher percentage of abrasion at 2.67% concentration. Then, both brands show the highest percentage of abrasion at 2.67% concentration, but the highest increase is at 1.33% concentration. It can be concluded that other than the concentration that has positive effect to the percentage of abrasion, the duration of exposure to the sample solution can affect the percentage of abrasion positively. Discussion of percentage of abrasion. Kilman told that some chemical can induce physiological effect on cell and cause cell abrasion, and this type of chemical known as radiomimetics. And the result of this study showed that when the mung beans were exposed to the sample solution, it induced the abrasions, and when concentration is increased, the percentage of abrasion for both sample solution are increased. Not only that, the longer the exposure had caused more abrasions. This means that there is some radiomimetic chemicals present in both extra exhaust and cucurbita brands. As the conclusion, the data analysis of the study shows that both powdered energy drinks may have toxic ingredient or chemicals because the effects after exposure to the sample solution to the mung bean. It had caused the reduction of growth rates, reduction of the thermometer index, and an increase of percentage of abrasion. For recommendation, more macroscopic parameters should be observed to get a better analysis about the effects of the sample solution on the mung bean. And this is the list of reference that had been used for this presentation. That's all from me. I would like to thanks to you for your attention.